In most organisms, non-coding genes are transcribed as precursors that undergo further processing. In the case of ribosomal RNAs, they are often transcribed as a pre-RNA that contains one or more RNAs. The pre-RNA is cleaved and modified at specific sites by approximately 150 different small nucleolus-restricted RNA species, called ncRNAs. ncRNAs associate with proteins, forming ncRNAs. While ncRNA part base pair with the target RNA and thus position the modification at a precise site, the protein part performs the catalytical reaction. In eukaryotes, in particular a ncRNA called ribonuclease, MRP cleaves the 45S pre-RNA into the 28S, 5.8S, and 18S RNAs. The RNA and RNA processing factors form large aggregates called the nucleolus. In the case of transfer RNA, for example, the 5 sequence is removed by ribonuclease P, whereas the 3' end is removed by the tRNAs. The enzyme and the non-templated 3-CCA tail is added by a nucleotidal transferase. In the case of microRNA, RNAs are first transcribed as primary transcripts or pre-mRNA with a cap and poly A tail and processed to short 70 nucleotide stem loop structures known as pre-mRNA in the cell nucleus by the enzymes DRASA and PASHA. After being exported, it is then processed to mature mRNAs in the cytoplasm by interaction with the endonuclease dicer, which also initiates the formation of the RNA-induced silencing complex, composed of the argonaute protein. Even ncRNAs and ncRNAs themselves undergo series of modification before they become part of functional RNP complex. This is done either in the nucleoplasm or in the specialized compartments called Cajal bodies. Their bases are methylated or pseudoretinolated by a group of small Cajal body-specific RNAs, which are structurally similar to ncRNAs.